Okay, thank you for joining us today, everyone. So today we have a webinar by Regina Martuscello. She's at Columbia University, and so her group published with us a few years back, and she's going to be talking about some recent updates in the technique that she published on, which was laser capture microdissection from post-mortem human brain. So she's going to be talking about her original publication and about some recent updates in the field. And towards the end, we'll be happy to answer questions. So hold those to the end, and we'll. All right. So Regina, we'll leave it to you.
Thanks so much, Ron. So I'm sure people will continue to trickle in as I'm talking, and hopefully no one, or if anyone has any questions at the end, we can go back and double check on a few things. So thank you for joining me today. Today I'm going to be talking to you, Ben, like Ron said, about laser capture microdissection in the postmortem human brain. And the original title of my manuscript was a laser capture microdissection of Purkinje cells in postmortem human cerebellum, a stainless protocol for RNA isolation and RNA sequencing. And anything that I'm talking about today, there is a full video protocol available on JoVE. The PMID is listed there. And anything I'm talking, you can see me doing in real time if something I'm saying is a little confusing and you want something more visual. So just very briefly, laser capture microdissection. What is this? So, it's a specialized microscope that has a UV laser. I have two of them on the right. You can see on the top is a Zeiss, and on the bottom is made by Leica. And depending on the scope, you can cut at anywhere from 5, 20, 10, 40x, whatever magnification that you need. And the main difference between these two microscope machines is where the collection cap is located for the tissue that you're going to be cutting out in the region of interest. And so the Zeiss is what I call a top-down model, where the collection cap is on top of your tissue and a puff of air pushes that tissue into the cap. And Leica uses a gravity model where you cut your tissue with your slide face down and it falls down into the cap. Both of these systems work really well. I have used both of them. They both have pros and cons depending on what you're doing. And we're going to talk about them a little bit more, but I highly recommend using whichever system that you have easy access to. So for the purpose of the publication that I put into JoVE, was to identify and isolate Purkinje cells from postmortem human brain to obtain high quality RNA for enriched Purkinje cell sequencing. And if you know anything about the cerebellum, you know that Purkinje cells are somewhat of a rare cell population compared to, say, granule cells, which will be in the thousands compared to the 1 or 2 Purkinje cells. Let's say if you're sequencing whole cerebellar cortex at once. And single cell sequencing for RNA-seq has become extremely sensitive, but it has not been able to identify Purkinje cells in post-mortem human brain. Flow sorting prior has worked a little bit for mouse and rat, and they are able to find somewhere around 1 to 5% in the cell population. But in humans, in post-mortem human brain, this has not been capable yet, or at least I haven't seen it. And so I wanted to design a protocol to specifically isolate high-quality RNA using no stains or dyes. And it's very important to note that this particular protocol is only as capable as the tissue morphology and identification of specific cell types without the use of dye. So if your cells are mixed in together, if they're the same size, if you need some sort of an antigen to differentiate them, this stainless protocol is not going to work most likely for you. So I want to just jump right into what is my protocol. So before you do anything, when you're doing RNA sequencing, it's extremely important to test your RNA integrity number, or your RIN, of your starting brain material. Because if you don't have high quality starting material, you're not going to have high quality final material. And I recommend doing this before you're ready to start any type of cryostat cutting or laser capturing. I usually go to the cryostat, I'll cut maybe 300 microns of section off my tissue, extract the RNA, test the RNA integrity. So you've done all of these things, you're now ready to start preparing for your cryostat cutting, for your laser capture. So the day before you do anything at the cryostat, you need to make sure that you clean all of your materials, your brushes, your roll plates, your razor blades, anything that is going to touch your tissue needs to be cleaned. And whatever containers that you're going to use for your tissue fixation, you're going to want to clean with depth seawater and RNase away or some other RNase product. And I don't recommend autoclaving anything prior to use because autoclaving can increase RNases. So you want to make sure that you clean everything, let it air dry the night before. And then if you want, you can also prepare your fixation solutions with RNase-free water and high-quality ethanol and xylene. But make sure you do this within 24 hours. Fresh is always absolutely best when it comes to ethanol. So before you're ready to cut your tissue as well, you're going to want to make sure that your cryostat is incredibly clean. And so you're going to want to take your RNase away product and 70% ethanol. You'll probably notice if you've done this before, if you spray that RNase away in the cryostat, it immediately freezes. So I recommend taking a Kim wipe. These are sterile. Spray it with your RNase away, followed by some 70% ethanol, and then you can clean anything in the cryostat. I do not keep my roll plate at -20, so I can clean that prior to putting it in the cryostat, as well as my brushes. And then your cutting temperature and your chamber temperature are going to be really important for the cryostat. So I recommend setting your chamber temperature to -20 and the object temperature, which is the cutting arm, at between -15 and -18. You're going to want it to be warmer than the chamber, but you don't want the chamber to be too warm. Otherwise, your tissue will stick to your roll plate, and then it's not going to be nice and flat. And so for this particular protocol and the tissue that I use, it is not embedded in OCT. So I have a fresh, flash frozen piece of post-mortem human brain. I will then cut a piece of this off, and I will keep that in the cryostat and return the rest of my tissue immediately to dry ice. And then I call the little metal piece that you put in the cryostat arm a cryostat chuck. And so you'll take this chuck and you'll add OCT on top of it until it's partially solidified. You want it to be a little bit shiny in the middle, and then add your tissue on top so that it doesn't melt into the OCT. And then you'll want to leave that in the cryostat for a various amount of time. So I want to just kind of go over really briefly what a workflow could look like for this. So because you're going to want to have all of your stuff ready to go, and maybe you want to UV your slides prior to use for about 30 minutes, on a typical day, I would come into the lab and I would immediately place my slides underneath the UV. After that, I would go and I would get my tissue and all of my cryostat cutting materials, and I would go down to the cryostat. I would get my tissue, put it on the chuck, leave all of the materials that need to come to temperature in the cryostat, close the lid, and leave that there. Go back to the lab, prepare my solutions, put them on ice if they need to be on ice. And by that time, your 30 minutes has probably passed, your slide is ready to go, your tissue is probably close to being at the right temperature, and then you can go back down and get the cryostat ready to go. So it's very important to allow your tissue to acclimate to your cutting arm and the cryostat. If your tissue is too cold, it's just going to shred. It's not going to have a nice, even cutting. It's not going to go into your roll plate. It's going to have all sorts of little shreds in it. If you have any shredding in your tissue is too cold. You got to leave it in there longer. I recommend cutting at around 14 microns and picking all of your tissue up at once. If you don't pick it up at once and you leave the slide in the cryostat, it's going to get cold, and then you're not going to be able to pick up the next piece of tissue. If you leave it at room temperature, the tissue is going to melt. And so it's really important to pick up whatever piece you're going to use all at once. And I highly recommend not letting the membrane touch the cryostat stage. It's very cold. It will detach the membrane a little bit from your slide. And you'll know that this has happened because it will have an iridescent appearance. And then under the microscope, it will be uneven, and then your cutting will be uneven, and then your tissue won't come off the slide. So once you're finished, and I want to state that steps 1 through 4 are going to be identical for any type of fixation and staining that you use for step 5. So I have a couple alternative staining and fixations, but everything prior to this for RNA is going to be exactly the same until you get to this point. So once you're finished with your slide, take it out of the cryostat very quickly, and then you'll immediately want to put it on ice cold 70% ethanol for 2 minutes. This is highly recommended by Zeiss, and this will help preserve your RNA in a light fixation. You'll then want to increase your ethanol concentration, 95%, 45 seconds, also on ice. After that, I move to a room temperature 100% ethanol for 2 minutes. And I do this to help bring the tissue up to temperature a little bit. And then we move into the xylene. And so 3 dips in 1 container of xylene, and then 5 minutes in a second container of xylene. You want to make sure you remove any water, any ethanol from the tissue before you let it sit in that second xylene. Otherwise, you're going to have some aqueous infiltration. And xylene is very important in this particular protocol because it sucks out all of the lipids in the tissue. And so you're going to get a really nice morphology, a really good visualization with this. And then extremely important, you must dry your slides upright. They must be leaning upright for at least 30 minutes. If you let them dry flat, everything is going to soak into the membrane. Your membrane's going to be too wet. It's not going to cut. And if you don't let it dry long enough and your tissue is too wet, it's not going to cut. If your tissue is wet and you put it in the freezer and then try to pull it out later, you may have ice crystal situations. So for me particularly, I would always cut my tissue on Monday, and then I'd have tissue for the rest of the week for laser capture. I have worked out this protocol to store them for up to 7 days in the -80. What you really want to make sure is that your slides are extremely dry, and store your individual slides in a 150 ml conical tube, 1 tube for each slide. I don't recommend putting them back to back. And then make sure it's a really tight seal. On the day that you're ready to remove these slides and go do your laser capture, you're going to do that 1 hour prior to your intended use. And do not immediately open your tube. What's going to happen is condensation is going to appear and you're going to get all this water in your tissue. So allow it to come to room temperature. Condensation will be on the outside of the tube only. And then once your tube is at room temperature, your condensation is gone. It's going to take about 30 to 40 minutes. You remove that slide or just open the cap and expose it to the air. I would usually leave it on my bench for an extra 15 to 20 minutes, and then I would go down to the laser capture machine and prep that for my collection. So before you use your machine, you want to make sure that it's clean. If it's not yours and if it's a shared machine, it's going to be really important to clean both the stage and the cap collection device. The last thing you want is someone's aberrant tissue getting into your collection device and messing up your sequencing signals. And as I mentioned, the Zeiss microscope uses a puff of air to send tissue into the cap above the slide, and the Leica uses gravity, and so your cap is going to be below your slide. So after you add your slide to your stage and your collection cap device, I highly recommend using a Zeiss opaque cap. It's this kind of white sticky cap, and it significantly enhances the visualization, at least on the Zeiss, because the eyepiece and the lens go through the cap to visualize the tissue. It will not do that on the Leica. However, the cap in my hands is able to preserve the RNA for up to 6 hours of collection. And I think that's really important and key, because if you're going to be sitting there for a long time and collecting, you don't want your tissue to dry out or to lose any RNA that you may have in there. So in my particular case, when I'm looking at my Purkinje cells, it's really important to visualize the tissue in the cerebellar layers at around 5 to 10x to identify where your molecular layer and your Purkinje cell layers intersect. And then I collect at 40x to specifically identify the Purkinje cells. And I have some pictures and some videos, and we'll be able to see what that looks like. I do want to note that each machine has a different name for the UV energy or the UV focus, which is basically the intensity of the laser. And so prior to collecting, make sure you test this in a region that is not going to be going into your cap so that you're not burning the tissue. If you see a dark black streak across your tissue and it's not cutting, this is going to tell you that your intensity is not high enough or your tissue is too wet for the cutting. So keep in mind that as time goes on, your tissue is going to continue to dry, and you may need to change this so that you don't have tissue flying off your slide. You don't want your energy or your focus to be too high, because you can also potentially shear your RNA. So in my published publication, this is one of the figures that I presented showing the importance of xylene in this stainless protocol. And so as you can see in panel A and C, this is what it looks like when you do not have xylene. I've indicated Purkinje cells with these black arrows, which are very difficult to see even under 40x. and the molecular layer and the granule cell layer, again, in the cerebellar cortex. And so when you add xylene, the visualization is night and day. You can see very easily even the white matter track here moving through the granule cell layer. You can see these Purkinje cells all along here in their line, the density of the granule cells, the sparsity of the molecular layer. This morphology is what allows me to identify these giant neurons right here for collection. And so, as I mentioned, the opaque cap through the Zeiss machine, panels A and B show you without and with the cap. And so you can still see the Purkinje cells pretty well here, but they don't look nearly as nice as they do when you go through that opaque cap. And when you use the Zeiss machine, they leave behind little circles with numbers to let you know how many samples you've collected. D and E is just an example of one of the cut and propels. And F just shows you an H&E of really how beautifully laid out the morphology of the cerebellum is. You're going to need something along these lines to be able to utilize a stainless procedure in identifying cells that you would like to isolate. So this first video here is very short, and I apologize, I took it with my cell phone. But when you're using the Leica machine, it's a touch screen, at least the machine I used, and they have what I like to call the laser pen. You touch the pen to the screen and it activates the laser. This is under the draw and cut function. And so all you have to do is put your slide on, open it up, make sure you can visualize really well, and then you just start cutting and collecting. It's very fast, it's very efficient, doesn't require really much of anything. And so you'll see I just touch the pen to the screen, I circle around my Purkinje cell, and it's cutting through the membrane and the tissue. And as soon as I'm done, the tissue is now gone. And that's what will happen on the Leica every single time. So compared to the Zeiss, which is a little bit more of a sophisticated program, it has a lot more buttons and options because of the puff of air that it utilizes. And so this is a computer program that will show you what's on the screen. And so here I'm identifying where the cursor is in the Zeiss. When you switch from 40x to 20x, the cursor will stay in the same place. I was identifying the Purkinje cell layer and the Purkinje cell 2U. So as we move out to 5x, you're going to want to adjust your exposure time because it doesn't really do it automatically. And you can see really nicely, I'm just going to pause that for a sec, the visualization here. And so the cursor is in the same place, right next to that Purkinje cell. You can really see this granule cell layer very well, this molecular layer, this white matter track going through here. And that's just what my cursor is now going to show you, the Purkinje cell layer in the video. And as you go back down to 40x, I think it's really nice that the cursor stays in the same place to help you identify what you're looking for. You want to increase your visualization, which is not essential for the collection, but essential for your ability to see. And so you'll select the line tool here, which is at the bottom of the screen. That is the Purkinje cell that we're going to dissect. And then we choose our center robo LPC. As you can see, there's a number of options on this one. I like the center robo LPC because it will put the dot right in the center, and then that puff of air will pop it right out after you hit the start button. And so as opposed to the Leica, it's not in real time. You draw your lines, your cuts happen, and then your tissue gets propelled up into your cap like that. And so I highly recommend blue light reflecting glasses anytime you're working at 40x, especially if your microscope is in a dark room. So with my particular protocol, this is some of the RNA examples that I can obtain. Section preps are what I call the testing product of my initial tissue prior to my laser capture. And as you can see, these are all pretty high in the high 9s. And then my RNA from my LCM is in the 8s, and I also got a 9.1. I was pretty happy with that. And the quantity of RNA I got is pretty substantial. It should work out pretty good with the total volume that I have at the end. And I included my PMI frozens. PMI is the post-mortem interval, the time from when the brain, from death till being frozen. And as you can see, they do range significantly in time, and they really did not have any effect on my particular RNA quality, but I do know that that can be a problem for people sometimes. And you can see my RNA readouts here with my 18 and 28S subunits. Everything looks pretty good. So when you're done with your laser capture, extracting RNA from your cap, you're going to want to use the RLT buffer that comes with one of the Qiagen micro or mini kits. At least that's what I use, along with beta-mercaptoethanol. This will help preserve your RNA while it's in the freezer. And so you'll dilute it at about a 1 to 100, so 10 microliters per 1 ml. And then you'll leave it in the cap. So you put it in the cap, close the tube over the cap, let it sit for a minute on your bench, gently vortex, lysis your buffer, and do a quick spin. And then repeat this process. Really make sure you get everything out of the cap. You don't want to leave anything behind. And then you can put this in the freezer. It'll be about 100 microliters, so not too small. And then you can remove them for your batch RNA extractions at a later date. So let's talk about some challenges in postmortem human brain, because there are many. You basically have limited to no control over the postmortem interval, or the PMI, or the freezing technique. And so, as I mentioned, PMI didn't really mess up my samples, but the way samples are frozen significantly alters your visualization capabilities. And so ice crystal formation is a real problem in postmortem human brain that's been in a freezer or not properly frozen. And so what'll happen is water, either from wet ice or the freezing process, will get into the brain and it will expand in the freezer. And as soon as that happens, it will create a honeycomb-like appearance as water expands when it freezes, and it will basically remove or damage all of your morphology. You will have absolutely no ability to really see the tissue. And this can also occur with repeated freeze thawing, which is why I recommend cutting off the piece of tissue that you want to use in the cryostat, leaving it in there to acclimate, and putting the rest back in the freezer for a later time. And same thing with the RNA integrity of your starting material. If it is less than a 7, you're going to have a hard time getting anything higher than that in your LCM product, because if it's a 6.5 when you start, it's going to be less than a 6.5 when you finish, maybe a 5. There are RNA kits that can help with degraded or partially problematic tissue, but I find the best starting material gives the best ending material. And if you're getting something from a brain bank, again, you really have very limited control over these things. So you can ask, have you tested the RNA integrity of your samples prior to requesting? And at Columbia University, we do have the New York Brain Bank and the ADRC, which does have tissue that is available for people. And we have world-class individuals freezing our samples. No ice crystal formation in any of my samples from the New York Brain Bank. So a couple of Microscope protocols tips. So in particular, if you're collecting Purkinje cells, you're going to need about 1,800 to 2,000 to have enough RNA starting material. And this can take anywhere from 2 to 4 days of up to 4 hours of collection. Sometimes I would sit there for 6 hours of collection. So just keep in mind, whatever the particular cell type of interest you have, see how much material you need, how much RNA you get out of it, and how much your kit needs. I used a low input kit, but there's also other Pico kits that can help as well. When you're using the Zeiss microscope specifically, make sure that it's on a vibration absorption platform. Otherwise, any collection cap that has a liquid in it and not an opaque cap will fall down on your tissue, and that will be the end of your experiment for that day. You'll have lost everything that you may or may not have been working on. When, again, I mentioned earlier, the focus or the laser intensity, make sure it's not too high. I've seen people use it at max level to really try to cut through their tissue. If your tissue is too thick, try cutting it a little bit thinner. If you can't cut it thinner, try letting it dry a little bit more. You do not want to use max laser on any of these machines. You will burn it out, you will burn your tissue, you can shear your RNA. So if your tissue is not adhering to your slide, which can be a huge problem sometimes, and that 30 minutes of UV exposure is just not sufficient, it's still flaking off, I find that when I'm finished picking up my tissue, if I put it at room temperature for maybe 15 to 20 seconds, that tissue will fully melt into the slide. And then when you move into your fixation and visualization steps, it will be much easier to have the tissue not fall off that slide. It'll really stick on there. And so I had mentioned that, my tissue is not embedded in OCT. It's fresh frozen. It's just a chunk of tissue. And OCT is not recommended for laser capture. So if you have samples that are embedded in OCT, you're going to need to remove that prior to doing laser capture. It'll mess with the machine. It'll mess with your sequencing. And you can do that by dipping the slide into ice cold RNase-free water. And that should get rid of all of the OCT. And I do just want to mention, my protocols here have not been tested with FFPE tissue. I do know people use it, but everything I'm talking to you about today, I just want you to keep in mind this is for fresh frozen tissue. And if you have not read the Carl Zeiss microscopy LCM protocols for RNA handling, I highly recommend it. If you're having a hard time finding it, I can share a PDF copy with the webinar individuals, and they can then disseminate it out to anyone who is interested. So there are a couple alternatives to a stainless protocol that Zeiss recommends and other people have used. I have tested this Crescel Violet once, and it works pretty well with the visualization. And I did look at the RNA, and it wasn't terrible, but it wasn't as good as with my stainless protocol. So as I mentioned before in the protocol page, everything from steps 1 through 4 are exactly the same, but you can implement this instead afterwards if you need to use something like a Crescel Violet. And I would do this 100% on ice, that you're not using xylene, keep it on ice the entire time. And so you always start out 2 minutes and 70% ethanol, about 30 seconds in a Crescel Violet solution, and then you'll want to remove that excess dye, otherwise it's going to be swimming in your 70%. And then just one dip in 70, one dip in 100, and then again, you want to dry it upright. And I know Zeiss and a lot of people recommend short drying times, but the shorter the drying time, the more liquid that's in your tissue and the harder it makes to cut. So I try to let it dry for at least 20 to 30 minutes before you go to the machine. And as well, I have not tested the freezing, but in the past, I have found that if you're not using xylene and you're putting them in the freezer, they don't quite last 7 days, maybe more like 2 or 3 days. And so you can also do something I called a shunted H&E protocol. So I don't use eosin in this protocol, which Zeiss does have a potential one that you can do that with. But instead, I use a filtered Harris modified hematoxylin. I find it's more of a quick stain hematoxylin, but Myers works as well. Gill, you can pretty much use whichever one that you want to. But that 2 minutes and 70% ethanol is key. You always want to do that when you're doing RNA. Do your 30 seconds of your filtered hematoxylin, and then remove that excess dye. Otherwise, your bluing agent is just going to be absolutely saturated. I have a recipe, but you're going to just want to use some ammonium hydroxide and some RNase-free water. And then 2 to 4 dips in 70% ethanol. And then you're going to want to dehydrate with increasing amounts of ethanol. So 95% followed by 100 anytime you're doing some type of hematoxylin. And then again, you want to dry that upright. So recently I have developed a new protocol, and this is for proteomic collection. So if you want to collect samples for protein extraction or liquid chromatography mass spec, really high intensity proteomic analysis, I have done this, and it works really well in cerebellar cortex, in the dentate nucleus of the cerebellum, as well as the inferior olive of the medulla. And so a couple of things to keep in mind with this protocol compared to RNA protocols. So you're going to need liquid chromatography or LCMS grade water for all of your staining and fixation, not the nuclease free. It's not going to be high quality enough. Your collection material is going to be a low adhesion 500 microliter cap that's filled with water. You're not going to want to use those isopaque caps because it's going to have a little bit of that protein in it, and it's going to mess with the proteomics. And then you'll want to use what's called a PET frame slide. These slides have metal with just the membrane in the middle. For RNA, I recommend the glass slides with the membrane on top. And then I always do my laser capture right after the staining. And then you want to use glass instead of plastic wherever possible. Okay, so for all of your cleaning procedures, instead of RNAs away, you're just going to use 70% ethanol. And so this protocol, again, is all on ice, and this is a shunted H&E protocol. Now, you're not going to do that 2 minutes and 70%. You go about 5 seconds. And then 30 seconds to a minute. Sometimes the dentate and the inferior olive need about a minute in that hematoxylin because it's got a lot of white matter and a lot of those synaptic nuclei. And then remove your excess dye. And then 3 to 5 dips in your bluing solution, which is just 40 microliters of ammonium hydroxide and 40 milliliters of your high-grade water. And then your dehydration series of 70%, 95%, and 100% ethanol. You can let this dry for 30 minutes to an hour, and it should be good for anywhere up to 4 to 5 hours for collection at your laser capture machine. And then all you do is just spin the cap down with the water, and you can hand that off to your proteomics core, if that's what I did, and then they'll be able to do the extraction and go from there. So this is kind of the entirety of my presentation of what I've done, what I can do, and what I've done lately. So if you guys have any questions, I'd be more than happy to answer any and all of your questions. So I do see that there is one question in the chat. Fresh frozen tissue, telecephalon, and single nuclei. To recommend LCM for obtaining tissue from different layers. Sure. Okay. So yeah, I would definitely recommend using laser capture to obtain tissue from different layers of the telencephalon for isolation of nuclei. That would be really great. And to remove OCT from the frozen blocks, what you're going to want to do is cut the tissue with the cryostat. And then once it's on your slide, dip that slide in ice cold RNase-free water. That will remove your OCT from the tissue, because there's really no way to remove it from the embedded tissue without melting it, and you don't want to melt your tissue. So just dip your slide in ice cold RNase-free water. But yeah, I absolutely recommend obtaining from different layers. So fluorescent samples. Fluorescent samples work really well, actually. So I do know on the Leica machine, they do have fluorocubes. And so if you have some type of a GFP, in the immunostain protocols, you know, they do work. I'm not sure if you heard all that about fluorescent samples, but yes, fluorescent samples work just great. You can use the fluorocubes on the Leica microscope. For immunostaining, it can work, but again, it's lightly fixed. So you're going to have to alter your immunostain. You're not going to be able to do antigen retrieval because your samples are just, it's going to be too lightly fixed. It'll completely shred the tissue. So it is possible, but I have not tested it specifically on my own. I keep saying to dry standing, vertical. What keeps your sample from sliding off? So not a dumb question. Excellent question, Davida. So once you pick up your slides on the membrane, they should kind of melt into the membrane a little bit. And then after you go through your ethanols and your xylenes, you're going to have the tissue is going to be adherent to the slide. I promise you it will not fall off once you go through that whole process. If it does fall off, then you definitely need to UV your slide before you go through that process. And you're going to want to make sure that the tissue is fully into the membrane. And you can tell that. So the tissue itself is going to look a little bit clear. So as it melts into the membrane, it's going to get clearer. You'll be able to see, it'll be somewhat opaque. If it's not, if it's just sitting right on top, then you haven't let it sit long enough. Hold it in the cryostat. Really make sure your tissue is on your slide. Take it out of the cryostat for about 10 seconds and then go into your fixation and visualization protocols. And I promise you it won't fall off. What do you do with the collected tissue after collecting it in case you need to go several days in a row? Yeah, so excellent question. So what I'll do is I'll collect every day and I'll only use one tube a day. And when I'm done with that tube, I will make sure I put some liquid in the top of my cap, collect my cells with that RLT BME buffer. I'll spin it down and I'll put it in the -80 freezer. And I have left my samples in the -80 freezer for months before I did RNA extraction. And so you definitely want to do that in a batched way and just keep putting them in the -80, get another slide. If you can't get another slide, if you want to use the same slide over and over again, you're going to be freezing and thawing it, and that's going to negatively affect your RNA. And so I recommend cutting a slide for every single day that you want to do collection. And then that should allow you to go for several days, keep that product in your -80. If you want it all together, you can put it all together later on. Just make sure you don't have too much of a volume for whatever RNA extraction kit you're utilizing. Is there a certain standard ISO to be followed? If you're talking about like for the reagents, like the ethanol or the xylene, you definitely want to use high grade, like 200 proof high grade histology grade ethanol. Same thing with the xylene. Fisher Scientific sells them at a pretty reasonable price, which is what we tend to use. Unless there's something else that you're asking me with the ISO standard. So in my particular samples, I can put up to 4 brain slices on a slide. And that's really going to be determined by how large your samples are that you're using at the cryostat. I recommend maybe I mean, you only want them to be a couple millimeters by a couple millimeters. You don't want a too large piece of tissue because the slide is only as big as the slide is with the membrane. So remember, the membrane is smaller than the slide. If your tissue is not on the membrane, if it's on the glass, I don't recommend cutting on the glass. But I could get up to anywhere from 3 to 4 sections per slide, which really helps when you have long-term collection.
Okay, so I think that's it as far as questions. So thank you everyone for the insightful questions. Thank you, Regina, for the excellent presentation. So we'll send out a follow-up email later with a recording of this webinar, so you can have that for your records. And yeah, thank you everyone for attending, and have a great day, everyone.